rise of multicellularity is deemed as one of the major transitions in the evolution of life. It seems that cells benefit from cooperation with other cells to survive and grow. However, the selective advantages that favor the evolution of multicellularity in organisms remain largely unknown. In Fujita Lab, our primary interest is to understand how multicellularity and its associated mechanisms help plants grow and thrive, despite their sessile nature. Utilizing the Moss viscometrium patterns, due in part to its amenability to be observed at single-cell resolution and rapid transgenic capabilities, our research tries to understand how intracellular polarity, cell fate, and cell-to-cell communication is established and maintained in plants and how it affects the external morphology and development of such said plants under favorable and stressful environmental conditions.
In Naramoto Lab, we are working on how plants regulate their morphology and the evolution of the regulation process. Plants develop a determined pattern of shape, with shoots growing upwards and roots downwards, and the shape of leaves and flowers are unique to each plant species. It is known that plant cells communicate each other for developing such a uniform pattern, with using plant hormones for signal transmission. However, precisely how remains largely unclarified. So, we are aiming to reveal the signaling process, from when and where the plant hormones are synthesized, and how it is transported, and what roles they have in plant morphogenesis. We also focus on the evolution of such processes by comparing the regulatory system and the roles in different plant species.
In our lab, we use a variety of plant model organisms, Moss viscometrium patterns, liverwort Markantia polymorpha, fern Ceratopteris richardi, and flowering plant Arabidopsis thaliana. We analyze these plants using molecular biology techniques like knocking out or overexpressing genes, investigating the site of gene expression by RNA sequencing and single-cell analysis, and live imaging of plant cells using fluorescent proteins and so on. For live imaging, we use high-end confocal microscopes capable of observing the precise localization and movement of cellular compartments. We also treat plants to various environmental conditions such as drought, heat, salt, and variable gravitational forces, utilizing special equipment that can grow plants under hypergravity or microgravity. By observing how plants react to different environments at the molecular level, and by comparing and combining the results from different plants and methods, we aim to answer questions pertaining to how plants adapt to their environments and define their morphology.
I chose to study in this lab because I am interested in plant evolution. A lab that uses several plant species is rare, especially plants across different phyla. For revealing the evolutionary process, it is important to compare between phylogenetically distant species. So I knew this lab would suit me. It proved me right with not only the plant species used, but also with the valuable and high-spec equipment used in the lab. Also, discussing with people with different research backgrounds gives you new insights, which sometimes leads us to a breakthrough in the research. Each member of our lab are working on diverse topics, and we believe that this environment will help us make the quality of the research higher. We are waiting for you to join our lab and study together in Hokkaido University.